All right, everyone. We are going to go through a guide for patterns of inheritance and distribution of traits. You should only really be here with me watching this video if you have reviewed the resources first, or this might get confusing because I'm kind of skimming over things and making sure that you understand at the top level, but I'm assuming that you've already tried to understand some of this already. So checkpoint 1 is to identify, define common terms related to genetic variation. This is basically, do you know the words on the right-hand side of the screen when you're taking the focus area? So just to briefly go over them. Genes and alleles. A gene is a sequence of DNA that codes for a protein that does a specific job. So like, for example, a gene for hair color. Whereas an allele is a variant of a gene. So if I'm talking about the gene for hair color, alleles would be things like blonde or brown or brunette or whatever they may be. And you always have two alleles on every gene. I'm sorry, two alleles of every gene, because on each you have two chromosomes at every location. And on the same location on both of them, you will always find the same gene. And again, those genes could be the same exactly, or they could be slightly different from each other. For example, if I have the allele for brown hair and then the allele for blonde hair, right? They would be found at the same location on the pair of chromosomes, but they might be very slightly different. So an allele is a variant of a gene. We write them as two-letter sets, the alleles that is. We also refer to this as a genotype, right? So we can write big A, big A, little a, little a, little a. This is in reference to the types of alleles that are being carried. There's always two because we have a pair of each gene in our body. So this would be, all the examples I was referring to earlier. The genotype is the actual genes that we have. So in this situation, right, we might have big A, if right here there was a big A and right here there's a big A. That would be the genotype. The letters refer to the genotype. The phenotype would be the physical expression of those genes. So if I'm making all the proteins for blonde hair, because my genes say to make those proteins, the phenotype would be actually having blonde hair. Sometimes those the genotype and the phenotype can disagree a little bit. So just remember that phenotypes are physical, right? The pH phenotypes are physical. Let me just make sure I'm recording here, because I'd be sad if I wasn't. All right. Recessive versus dominant. So if I have my genotypes here, in this situation, we're saying that this parent has something like this. So we write with lower letters to express a recessive trait, and then we write big letters to represent a dominant trait, right? We're saying that each of them gave one of those alleles, like this, and that would make this child have inherited something like that. But because they have the dominant allele, it is always expressed in the phenotype. This is one of those situations where their genotype has both the recessive and the dominant trait, but because of the rules of how this works, the phenotype always is a little bit different than what the genotype might hint. So knowing that if this big A is present, it will always express itself because it's dominant is very, very important to understand. Here's another example again, right? Notice that we have the big A, the little a, the little a, and the big A, little a. These are exactly as you would expect, but notice because the big A is dominant, that is still expressed in the phenotype. Homozygous versus heterozygous keys in on the fact that the word homo means same. So notice that in this case, they're both big A's, and in this case, they're both little a's. Homozygous is in reference to that the letters are the same, either both big or both little. Zygous just refers to the alleles themselves. Are the alleles the same or not? The other term, other than homozygous, would be heterozygous. Hetero means different. You'll notice in this far right chromosome pair, we have a big A and then a little a. They're not the same. One's big, one's little. So we call that heterozygous. The question being, if the one on the far right is heterozygous, how do we distinguish between this one with the big A's and this one with the little a's? They're both homozygous. The simple answer, we just simply say dominant or recessive after it. So in this case here, we say a homozygous dominant. In this case here, we say homozygous recessive, because they're both recessive, and this one, they're both dominant. There's only one heterozygous, so that makes our lives easy. Okay, checkpoint 2. Can you use computational thinking and patterns to predict how traits are passed from parent to offspring? This is basically Punnett squares. Can you do a Punnett square? So in a karyotype, right, when we look at something like this, each parent, let's say this person has a big A and a little a, right? Only this side has the big A, this side is the little a. You only pass one of these two chromosomes to your child. We split all of our DNA in half, and we give half of it to our child, half of each of the chromosome pairs. And it's random which one they get, whether they get that little a or whether they get that big A. So a Punnett square helps us predict which our child will receive. So in this case, let's say we have a dominant allele. Let's go like this. And the dominant allele, let's say, is a big A, and let's say that codes for blue hair. And then we have a recessive allele, let's say it codes for brown hair. I like to write out all the possible genotypes. So we have a big A, big A, big A, little a, or a little a, little a. And then I like to write out the possible phenotypes, which we have, you can have blue hair, or you can have brown hair. Wow, my writing is atrocious. Okay. So, and then I like to link to which one goes to which, right? That's really bad. I gotta erase that. That's bad. All right. So I'm gonna write blue twice, and I'll show you why in just a second. There we go. So this genotype leads to that phenotype because it's the dominant trait. This one also leads, because it's heterozygous, to the dominant trait. The only way to get brown hair is if you're homozygous recessive, right? So I like to write all that out to make things clear for me. So what this is going to do, let's say I have a parent who has blue hair and is heterozygous. So that would be their genotype. And let's say they have a child with somebody who also is heterozygous. You would write it like that, one for each of these rows. And then all I do, in order to predict what combinations they could get, their children could get from their parents, I would pull this one down, just like that, and then I'd pull this one down, and then I'd pull these across, and pull these across. So I would write it like, let's do a different color, let's do the blue. So big A, because I'm pulling this big A down, and then this little a I'll pull down. Then I'll pull this big A across, this big A across, little a, little a. So now I know that my possible genotypes for my children, 25%, because it's 1 out of 4, would be big A, could be big A, probability. 2 out of the 4 boxes have big A, so half of them will be heterozygous, and then another 25%, or 1/4, would be little a. So in other words, I could say I have 1 big A, 2 big A, and 1 little a. This is what's known as a ratio. A 1-to-1 ratio is very typical for the genotype for heterozygous individuals, right? But if I were to talk about genotypes, things change a little bit, because phenotypes. What phenotypes am I going to get? I'm just going to write out what I got over here again to have it be a little bit clearer. So if we're talking about phenotypes, you'll notice that these three all have the same phenotype. We have 1 of the big A, and we have 2 of the big A, which all have blue hair. And then we only have 1, that's little a, that is to say brown hair. So the phenotype ratio, the physical expression of these genotypes, would be 3-to-1. That is to say 75% to 25%. And that's how basic Punnett squares work, right? This is an important step to write that out, and also always drawing things out. So if I were to give you any kind of combination, say the parent is one genotype, the other parent is this genotype, you should be able to figure out and predict what percentage of their children would inherit which genotypes. Things get a little bit more complicated than this. If I go back to the karyotype, instead of just writing big A, I could also indicate which chromosome they come from. So if I was saying it's something on chromosome 1 and there's a gene here, and this one is big A, this one's little a, I could write this as 1 big A, 1 little a. Just this is telling me, of course, what chromosome it's on, and this is indicating which allele that chromosome carries. I could do the same here, right? Let's say it's Z here. That's a bad one. Let's go T, big T, little t. I could write 15 big T, 15 little t, right? And this would be my genotype, and I'm just expressing which one it is here. So when we talk about the sex chromosomes are the ones that determine your sex. XX would be female, and XY would be male. I can indicate, like exponents, what genes I'm talking about if they're on there. So if we go back to our sex-linked crosses, this gets a little bit hard because there's so many possible combinations. If we're talking about color blindness, which is found on the X chromosome, let's open this up. I could have a female that is XX, and the let's say we're going to use R for color blindness. The dominant allele, let's say the big R, is normal color vision, and the little r is no color vision. So I'm going to write that down just as a little note to myself. So the three possible combinations of genotypes for females are big R, right, normal vision, big R, colorblind, normal vision, and then little r, colorblind. Where it gets tricky is with the males. The males can be XY, right, or they have to be XY, but they only have one X chromosome. So that X can either have the big R or it can have the little r, but the Y has a different set of genes entirely, so they just don't even factor into anything. It's just, do you have it or do you not when it comes to males? So these are the possible genotypes, right? The genotypes I've written out here because there's so many of them. Then the phenotypes linked to this. This is normal, or this is vision. This is vision. But this is no vision. And for males, it's simple. Vision, no vision. So let's say we have a colorblind female, XX, and both those X's have the color blindness allele, and a normal color vision male. So let's give him the big R. I'm going to put the X's and the Y's down first because I find it easier to take one thing at a time. So I'm dropping these X's down, and then I'm going to pull these X's across. Okay. And then I'm going to transfer the exponents. So big R coming from here. The Y, of course, doesn't have the allele, so I don't bring it down. Then that little r, I put on the X's coming down. So notice that all the female children, let's change colors again. Let's go orange. All the female children, which are these top ones here, are carriers. They're heterozygous, but they have normal color vision, right? They match this right here. The male children, on the other hand, both inherited the little r from the mom. So if we look here at this combination, the male children are colorblind. So if I were to ask you what percentage of the male children are colorblind, you would say 100%. If I were to ask you what percentage of the children are colorblind, you would have to say 50%. Notice that the question changes if I ask you male or female. If I ask what percentage of the female children are colorblind, right, you'd say 0%. So all you have to do is make sure you're paying attention that there's 4 squares. How many of them would be colorblind if I'm saying how many children would be colorblind? But if I'm talking about male or female, there's only 2 squares. And so I'm asking how many of those squares, in this case of the male children, would be colorblind. So, okay, that's sex-linked crosses and normal crosses. Always draw out your Punnett squares. Always write out your alleles, what's dominant, what's recessive, the genotypes and the phenotypes linked to them. It'll make your life much easier, even though it makes you go slower. So checkpoint 3 is to describe how population genetic variation is caused by mutations, gene flow, and sexual reproduction. I'm going to go through this fairly quickly. But basically, mutations, all variation is caused by mutation. right? There's a mistake made in the in the gene sequence that leads to a different phenotype of some kind. Usually mutations are bad. In this scenario, right, the normal gene sequence, which is on top, which is that's how it goes. But let's say there was a mutation here in this T, it changed to a T instead of an A, and that resulted in an embryo that turns out to be a red circle instead of the normal yellow. Again, if this is the typical environment, the mutation is going to suffer far worse. And so this mutation is going to die out. However, let's say the environment changed, and this same thing kind of happened. Suddenly, the normal is not helpful. And if it hadn't been for this mutation, everyone would just die and go extinct. Luckily, though, we happen to have this weird mutation that was harmful in the past, suddenly now is helpful. Now, who would survive better? Well, the red one would, and then the normal gene sequence would kind of die out, and the mutated would now become the new normal. So mutations, answering the question, how do mutations cause genetic variation? Most of the time it hurts things, but sometimes it creates a slight benefit for the environment, or at least a neutral one, so you get differences in how the population looks. Gene flow is the idea that different populations can interbreed and bring, like, this population here has a different genetic variation than this side, but if this one migrates and travels over here, suddenly there's more genetic variation in the population than there was before, right? If there was a mountain in the way of these guys, let's say all these guys just had the same alleles, and all these guys had the same alleles. If there's some migration, suddenly we're introducing these new alleles, the big A's, into this population, which may or may not be helpful depending on the environment. So gene flow causes genetic variation because it shares genetic variation from other locations. Sexual reproduction causes genetic variation because you get reshuffling of different traits. You get new combinations of traits that didn't exist in the parent generation. For example, you notice here, with the pink square, it has a black outline, and then this one is blue with a light blue. You'll notice that this variation didn't exist before, but because they had sexual reproduction, they could mix up their traits, we get a new combination of traits that wasn't there before, and same here. So that increases genetic variation by reshuffling all of the traits together and getting different variants than was there before. All right, and that's it. Hopefully that helps. See ya.